Welcome you all. Myself, Miss V.B. Mani, working as Assistant Professor at Satara College of Pharmacy, Satara. In this video, we are going to see about estimation of the quinine sulphate by FLUORIMETRY. The aim of the experiment is to estimate the amount of quinine sulphate present in the given sample by FLUORIMETRY. After performing this experiment, these three types of the objective will be achieved. So the first one, student will understand the principle of fluorimetry. Student will understand the handling of fluorimeter instrument, and student will learn about how to prepare the dilutions of different concentration. So, these type of the glasswares, chemicals and instrument will be required before going to proceed for this experiment. Now, we have to see the principle involved in the estimation of the quinine sulphate by FLUORIMETRY. A large number of the substances are known which can absorb ultraviolet or the visible light energy. But these substances lose excess of energy as a heat through collision with the neighboring atoms or molecules. However, a number of the important substances are also known which lose only the part of this excess energy as a heat and emit the remaining energy as electromagnetic radiations of a wavelength longer than that of absorb. This process of the emitting radiation is known as luminescence. In a luminescence, light is produced at the low temperatures. Therefore, the light produced by this process is called as light without heat or cold light. There are two types of luminescence. First one, fluorescence, and second one, phosphorescence. When a beam of light is incident on a certain substances, they emit visible light or a radiation. This phenomena is known as a fluorescence, and the substances showing these phenomena are known as fluorescent substances. The phenomena of fluorescence is instantaneous and starts immediately after the absorption of light and stops as soon as the incident light is cut off. When we are going to call the phosphorescence, when light radiation is an incident on a certain substances, they emit light continuously even after the incident light is cut off. This type of the delayed fluorescence is called as a phosphorescence, and the substances are called as fluorescent substances. Material exhibiting fluorescence generally re-emit the excess radiation within 10 raised to minus 6 to the 10 raised to minus 4 seconds of absorption. On the other hand, material exhibiting phosphorescence re-emit the excess of the radiation within 10 raised to minus 4 to the 20 seconds or longer. Thus, the lifetime of the phosphorescence is a much longer than fluorescence. Some examples of the fluorescent material are quinine sulfate, reserpine, aspirin, thiamine, etc. Quinine sulfate in an acidic media exhibit fluorescent property. So, for the measurement of the fluorescent property, fluorometer instrument is employed. Now, we have to see the procedure. There are the two parts. So, the first part of the procedure is procedure for the constructions of the calibration curve. And the second part of the procedure is procedure for the preparation of the tablet solution. So, one by one, we are going to see about the procedure. So, the first one, procedure for the constructions of the calibration curve. Into a series of 10 ml flask, transfer 1 ml, 2 ml, 3 ml, 4 ml and 5 ml of standard solution of quinine sulfate. The concentration of the standard quinine sulfate is 1 microgram per ml. Then dilute the series of flasks containing 1 ml, 2 ml, 3 ml, 4 ml and 5 ml of quinine sulfate up to the 10 ml with 0.1 normal sulfuric acid solution. So these are the diluted series of the flasks containing quinine sulfate and the 0.1 normal sulfuric acid solution. Then select the proper excitation and the emission filter, 365 nanometer and 459 nanometer. Then measure the fluorescence with the help of the fluorometer instrument. Enter the values in a tabular form and form the data, that is from the fluorescence and the concentration. Construct a calibration curve. Now we have to see the second part of the procedure, that is the procedure for the preparation of tablet solution. Weigh 20 tablets and reduce to fine powder with the help of the mortar and pestle. Then weigh accurately a quantity of the powder equivalent to 100 milligram of quinine sulfate. Then transfer into 100 ml capacity of the volumetric flask. After that, pipette out 75 ml of the prepared 0.1 normal H2SO4 solution and transfer into the same capacity of the volumetric flask. Then sonicate for the 5 minutes for mixing the content thoroughly to dissolve the drugs. Then dilute up to the 100 ml with the help of the same solvent, that is 0.1 normal H2SO4 solution, and filter. Make the label on that volumetric flask as tablet stock solution. Then mix well and filter with again the Wattman filter paper. Pipette out 1 ml of the filtrated tablet stock solution and transfer into 250 ml capacity of the volumetry flask and dilute the solution up to the 250 ml with 0.1 normal H2SO4 solution. The this is the dilution number 1. Make the label on the volumetric flask to avoid any confusion between. Then pipette out the 1 ml from the dilution number 1 solution and transfer into 10 ml capacity of the volumetric flask. Then dilute the solution up to the 10 ml with 0.1 normal H2SO4 solution. So this is the final diluted tablet solution. Make the label onto that volumetric flask as a final tablet solution. Then select the proper excitation and the emission filter and measure the fluorescence with the help of the fluorometer instrument. After that, calculate the concentration of quinine sulphate from the calibration curve. So this is the observation table. In the first column, this is serial number. In the second, name of the sample. In the third column, concentration of the solution. And in the last column, that is fluorescence of the solution. So, from the 1 to the 5, that is the standard quinine sulphate solution. And number 6, that is quinine sulphate final tablet solution. You have to write down. Then concentration of the solution you will have to determine, and how to determine the concentration of the solution we have seen in the previous video. For that you will have to refer this video for the determination of the concentration of the solution and fluorescence value which is obtained from the fluorometer instrument. You will have to write down here. And then find out the concentration of the quinine sulphate final tablet solution from the calibration curve. Then you have to write down the results and conclusion. So these are the references. Thank you all of you for watching this video. Thank you once again.